Hello, everyone. Welcome to the SayoStudio Science Podcast. It's good to be back. We have some new studio updates before we jump into another exciting scientist interview. So kind of just another recap of what we've been working on, what makes us love what we do. Nicole, do you want to start with some other
Sure.
subjects?
Yeah. I'm so happy to be here and have all of you with us today. I was going to start off with kind of my favorite project that we've been working on recently that I haven't really been working on. You and Anna have been, and that's the fish hats project and other critters. And so that's a fun one that we're doing for our local city water education program. I guess that's the quick description of the department. And so they do a lot of outreach and hands-on programs with kids. And so we got to create these hats for kids to color that they print out and then they put around their heads. So Claire, do you want to say which animals you've been working on for these fun hats?
Yeah. So we've gotten to do a variety. We started out with the peamouth fish. I think I'm saying that right.
Yes, it's very unique, and I didn't know about it until we learned about it through this project. So
Yeah, and wasn't they're just like their spawning season? Am I saying
yeah,
that right?
I guess it's only like 2 or 3 days. We should look up that information
Yeah.
about, that when they come to spawn back in very specific places, kind of like salmon. And so, the city of Bellevue is one of the few places that has, like, a live stream capture of the fish coming back to spawn, so
That's
I
really
think we
cool.
just missed it. It just happened, so
Yeah, just last Saturday.
yeah.
Yeah, and so we started out with that, and then we got to do some river otter hats, which was really fun. And then I'm gonna start working on a beaver hat. And then finally we get to do kind of a variation, which is a crown. Is that with plants, Nicole? Is that right?
Yes, it'll be
Okay.
kind of the ideas that we need to solidify are that it'll be a stream edge, kind of
Oh,
plants at the edge of a stream, and maybe show some of the insects above and below. We'll have to
oh,
figure out those
cool.
details. Yeah.
I haven't even thought through that, so I can't picture that as a crown on my head, but I'll have to start thinking about it.
Yeah. It'll be a fun challenge.
Yeah.
And also keeping it simple enough that it'll be fun to color for all age groups. So,
Yeah. Yeah.
yeah. But, yeah. Okay. So, what's the next one on our list? I'm trying to think.
I think maybe the giant squid?
Yes. Yeah,
Yeah,
we've had a lot of fun, more natural history projects recently. So,
we have.
yeah. And
Yeah,
that
this
one we're
is
just getting started on.
yeah. Yep. Tell me so this is really I think mainly just the squid, but it's going also to be a giant display.
Yes.
So that's probably one of the most technically challenging pieces. What were the
As far as the
dimensions?
size. So I can't remember the height, but the length of the wall is going to enable it to be life size, basically. So it's 40
Okay.
feet wide,
Okay.
which is kind of the size, the middle range between male and female giant squids, from what I've
Cool.
read initially. So yeah, it'll
Cool.
just be a really fun, huge species feature of giant
Yeah.
squid.
And you mentioned before having done a mural of similar dimensions and the printers wanting to do it in like panels. And so, working with the color art to get them all the files that then line up to be able to put it on the wall.
Yeah, I'm really curious to hear what the print shop that they work with, how they want to go about it. So,
Yeah. Yeah.
yeah.
But that part's kind of fun when it comes to
Mhm.
exhibit work, which we love to do and we'd love to do more.
Yes.
Yeah.
Yeah. And then what is the third one we want to talk about? Um,
Oh, do we want to talk about working with the 3D microscopy images?
oh, yeah, because that's actually a fun intro to
Yeah.
our today as well. So, Yeah, do you want me to describe that a little
Yeah,
bit?
you'll be
So
working on that one too, so.
yeah, so it's been kind of a slow rollout, but for CZ Biohub, we're going to be taking some of their data where they're looking at like real time um, locations of cells and how they might change, like say in an entire organism, like this one they're looking at, I believe, is a zebrafish.
Yeah.
And so it'll be a fun challenge to process all of the data frame by frame of all of these different cells that they've compiled from their 3D microscopy data. And so then we'll be working with them to figure out which cells they want to highlight and why, and working on telling a story that way. So we're just figuring out kind of all the technical details. So, yeah.
Yeah.
And that is a different department than the guest we have on today, who
Yes.
is also from CZ Biohub and focuses on the individual proteins in a cell. So we're excited to have him with us in just a moment. All right, we're ready. Okay, I'm getting in the zone. All right, welcome everybody to the SayoStudio Science Podcast, where science, storytelling, and art collide. I'm really excited about today's episode because we're diving into the hidden world inside our cells and how science and art can work together to reveal it. Excuse me. Joining us is Dr. Manuel Leonetti, who leads a research group at the Chan Zuckerberg Biohub. His team is on a mission to map where proteins live inside cells using cutting edge microscopy. And they're sharing that treasure map with the world. We even had the chance to turn some of their findings into a hand drawn animation recently, which was really fun. Manu, thank you so much for being here with us today. Um, to get us rolling, I have a really basic question. So, reading through all of your history of the work that you've done, so it looks like you started out as a chemist in chemistry, and then you continued your education in molecular neurobiology. I tend to kind of lump you in as a biochemist. What do you call yourself? What?
Yeah, you're right. Well, first of all, thanks so much, Nicole, for the invitation, and it's great
Yeah.
to get connected again. And yes, you're absolutely right. I trained as a chemist. I think fundamentally, I started as what's called a structural biologist, which is somebody who's really interested in how different pieces come together to build life. I think this is what interested me in chemistry, because chemistry is a lot about understanding how atoms are coming together to build molecules. And once we know the rules of how atoms interact, we can design new ways to create new molecules. And in biology, there's a lot of the same questions. And structural biologists have been asking these questions classically at the level of uh, proteins, and understanding how different atoms, or the building blocks of proteins, which are called amino acids, how they're coming together to form proteins. And I think very much on the same journey, but keeping zooming out
Hmm.
at a higher level. And today we're looking at an entire cell, and really trying to understand how the structure of the cell itself is organized, as all these proteins now, which are really the building blocks of the living, are coming together, interacting with each other to form life. So yeah, I guess throughout my journey, I've been asking very much the same questions, which is how things are built, and especially
Hmm.
built as ecosystem of these components that interact with each other. But zooming out from the atomic to the molecular to now the cellular level.
I'm I'm curious because I of course never went nearly as deep as you, but I mean I think some of what you said that you enjoyed in that summary is also what I really enjoyed when I started out in biochemistry, and I really enjoyed picturing the way different things come together. Like I love just like learning about the enzyme pocket and how, like the structure function sort of thing. And then I think that you're one of the reasons why we have so much fun working with you is because it seems like you're very visual. We've talked before about the Lego analogy, which I'm excited to hear more about. I'm kind of curious. Do you tend to kind of visualize, like, do you picture in your head these things coming together in the cell landscape?
Absolutely.
Yeah.
And I think as a science, cell biology is deeply visual. And
Yeah.
of
Hmm.
course, cell biology is a science that that was born out of the invention of the microscope, centuries ago. But for the longest time, and it's still true today, a lot of what we do is just looking at things
Mhm.
through the lens of more and more complex microscopes or systems now. But
Hmm.
cell biology is a science that is visual in its
Mhm.
foundation.
Mhm.
And for the longest time, and what I love about it is that for the longest time, what people were doing is they were looking into cells using, again, different kinds of tools, and just being drawn by what looked cool or surprising. They're like,
Yeah.
what is that weird thing there? There must be something really interesting about that. And really, that's how people used to prioritize the questions that they were asking. And I think
Hmm.
that's such a great way to discover the world and do science. Is like, let's look at the weird things. And
I
Yeah.
never
behind
thought
every
of that
weird thing
before.
is a very often a really cool discovery.
Yeah. I never thought about how much science is driven by the weird thing and trying to investigate why it's weird.
Right.
So I have a question along that line again before we get into all of the incredible details about your science. So just more on like a basic level, you're using a lot of fluorescent tags, right? To visualize uh, in a 3D microscopy way where proteins are localized, right? So that this leading question is, so where do you think artists, illustrators come in, and are they needed when you have all of this visual information from a microscope?
Yeah, I think that's a great question. And the answer is they're absolutely needed because a lot of the um, um, what we capture with our experiments is just the tip of the iceberg. We have a very fragmented set of information in the observations that we make, and sometimes that leads us to think that cells or the objects that we're looking at are much more simple than they really are.
Mhm.
Um, Something that is absolutely mind blowing is how busy the inside of a cell is. Sometimes people represent the cell as this kind of like, bag of liquid with a few things floating around, but it's absolutely not like this.
Yeah.
It's like this super, you know, high density
Yeah.
right? Fluid where all these different objects are really shoulder to shoulder with each
Mhm.
other. Most of them are proteins, which is really it's something that's very hard. It's so densely packed, I guess, that we don't come across these kind of examples in everyday's life. Maybe in a football game, you'll see something like that. But some artists, and there's a famous cell biology artist, David Goodsell,
Yes, he's
who
one of my
is
inspirations.
right.
Yeah.
He's doing this absolutely fantastic watercolor,
Mhm.
realistic watercolors of what the inside of cells look like. And I think something
Mhm.
that comes from his art is really this realization of how busy and complicated things
Mhm.
actually are. And I think that's the power of art, which gives us an intuitive view on something that is a little bit complex and to some extent counterintuitive to
Mhm.
realize when we look at just the very fragmented little measurements that we do in our experiments.
That is a great description. Claire, do you want me to ask another question, or am I have one more, but I just want to
Yeah,
give you a
please
chance.
go
Okay.
ahead.
So, I guess most of our listeners, you could, well, I shouldn't say you could, but I think they self-identify as science nerds. But still, just to give a baseline. So if we just
Go
wanted to give a
science nerds.
description of like proteins, like, it's not just the stuff you put in your smoothie, right? It's the building blocks, as you've said before, of our
Yeah,
cells and of. So do you want to give a better description of all the jobs that proteins, I mean, you can't tell us about all the jobs, but how important proteins
100%.
are.
They
Yeah.
are the molecular building blocks. As you said, the analogy that I use often is that of a Lego brick.
Uh-huh.
Proteins are really the Lego brick of the living. There is about 20 different flavors of proteins that make all of our cells. 20, sorry. There's about 20,000 different
Yeah.
flavors of proteins
Okay.
that
I was
make
gonna
all
say
of
I
our
didn't think it was
cells.
just funny.
So,
Okay.
and each cell type is going to use a different set out of this 20,000 total number of proteins. And really, very much like, Lego bricks are coming together to form bigger structures and interacting with each other. Like the same brick could be part of a lot of different assemblies. The same is true with proteins. They're really like the busy molecular players that make life possible. So what
Mhm.
do? They create the structure of cells. So
Mhm.
they create, for example, the skeleton that makes all of our cells, that gives our cells their shape.
Mhm.
They create forces that what allows our muscles to contract, for example,
Mhm.
and they perform all the chemistry. So all the enzymes that help us digest our food. For example, they're all proteins as well.
When
Um,
when you were mentioned just how densely packed a cell is in reality, the skeleton was one of the first things that came to my mind because, depictions of it sometimes can be very neat but that's one of those things when you see it in a microscope, it's just wild.
Absolutely,
Yeah.
yeah. And you're right. The skeleton of our cells is made from like little arrays of proteins that are making these very, very long structures
Mhm.
that really give the structural scaffold and the structural integrity to all of our tissue, essentially. And actually, and on that just remind me of something that's interesting at the intersection of art and visualization
Yeah.
and scientific understanding. Um, um, there is a part of the skeleton of cells, so that's called the cytoskeleton. That's called a microtubule. It forms these very long kind of cables inside of cells, and these cables are used by some proteins to move things around in our cells. So if you think of a neuron, for example, there are neurons in our brain that extend all the way to, our spinal cord. In my case, some of the neurons are cells that are like more than a meter long, right? It's very, very long distance to travel. For things to travel that distance, you need essentially little motors, right?
Mm,
Kind of like you can think of them as little cars that are gonna essentially drive along these microtubules to go from point A to point B. It's a beautiful process, and scientists have studied that process by taking like a few static snapshots of what all of these molecular machines look like. But
Mhm.
really, I think the real understanding of what happens is when artists took all of that information and start building movies of what the
awesome.
entire dynamic process might actually look like. And there's some few beautiful examples on YouTube,
Yeah.
etc. Right? And I feel like, especially when students see that, the power of this visualization, the beauty of it, because there's one of this motor so this motor protein that looks like something
Yeah.
with two legs. That I'm sure you've seen that
Yeah.
movie before, right?
I've done
Um,
an illustration of that, and that was, like, 20 years ago,
Yeah.
and it was yeah, something that really made me feel like, "This is why I love doing this. This is so cool, the
Absolutely.
little feet moving along."
It's all right. It's amazing, and it's so inspiring also to realize, the yeah, the molecular life of our cells look very similar to some of the things that that we interact with in the real world. But anyway, my point is, artists have this superpower
Mhm.
of giving us an intuitive, bringing us closer to these very difficult technical concepts, but also making us realize that we can use our intuition to understand these difficult things way more than we think. Right?
Yeah. That's something that Nicole and I always love to imagine that our role is probably much greater than it is in reality. But when we get to work with so many incredible scientists and really just bring, you know, visual inspiration in any way we can, we're like, I wonder if we've ever, you know, made a scientist approach it in a new way or think of it in a new light or bring in an analogy that they wouldn't have otherwise. So yeah, Nicole and I have that conversation often. That's our dream. Yeah.
But you know, a lot of our intuition, even as scientists, to
Mhm.
have to have new ideas, right? To generate new hypotheses, etc. I mean, we use the same intuition that we use in everyday life. At least I do, right? And so
Mhm.
sometimes being able to visualize something is that gives us the power to again like use that intuition that we know and trust and have developed through all of our interaction with the world in our own scientific questions. Um, for me, but maybe as we were discussing before, I'm a very visual person. If I can visualize the biological processes that I'm studying, it gives me better ideas of what you know what what to look at next or how to approach a problem.
I have a two-part question based on what you just said. So one, I want to hear from in your words about your current research about really looking at protein location in a big way, not just looking at one or two proteins, but looking at, thousands at a time and how exciting that is and why. But then before I forget, I also
Mhm.
want to make sure that I ask, so like as you have been studying this and figuring out techniques so that you can look at all of these proteins, have you found things that have been surprising? Like, I did not expect it. Have you found the weird in your results?
Oh yeah, yes we have. But maybe to your first question, you're absolutely right that the one of the big frontiers in cell biology is to be able to take global pictures of how an entire cell is organized. So traditionally, we've had very great tools to look at one or two proteins at a time in isolation, right? And learn a lot about the very small number of proteins. But there are billions of proteins that together make a single cell. And so if we want to understand how all of these system, right, at the level of the entire system of how all of this is organized, this is where our tools start to become a bit more limiting, and this is where we have to invent new technologies. Um, and so that's something that we're doing in the lab right now. And actually, to do that, we're starting to move away a little bit from the microscope and use other methods. In our latest work, we use a method called mass spectrometry, which I'd be happy to talk more about. But that allows us to take more global sensors and more global snapshots of all the proteins that are in a specific cell at a specific time, and to a large extent, where these different proteins are localized in terms of subcellular compartments.
I feel like I should have known. I probably we must have talked about that, using mass spec. But yeah, I would love to hear more about that because I have to admit my understanding of mass spec is like what they made us do in general chemistry like a long time ago. So I'm not really I don't know how you would use it for something like protein localization.
Um, Mass spec is you might be familiar with DNA or RNA sequencing.
Mhm.
Um, Mass spec is a very similar approach. It's kind of like a sequencing approach for protein, and it allows you essentially, if you have a sample, to identify the list of all the different proteins in that sample, and also their abundance. Okay, so you're going to
Okay.
be able to say like, oh, I have like 3 times more of this than that,
Yep.
so on and so forth. The key advantage that we're capitalizing on is that cells are extremely compartmentalized, which is that different functions in cells happen at the level of specialized compartment. There's a part of the cell, for example, called the mitochondria that you might be familiar with, which is where most of the cellular energy gets made. So all the energy production, the same as in a power plant, happens in one part of the cell. Um, um, all the dealing of genomic information happens in the nucleus, which is a different compartment. There's a recycling plant in our cells called the lysosome, where like dealing with all the trash and so on is going on. So again, the fact that cells are so internally compartmentalized is a huge advantage for us, because if we can take these compartments apart, which we can, and then apply mass spec to identify which proteins are in which compartment, we can get a fairly global but also detailed spatial map of the cell. And this is essentially what we've done in some recent work.
So I have a question. And this might be naive, but does the compartmentalization happen before the mass spec?
Yes, the compartmentalization is going on in the cell itself.
Yeah.
He's talking about the
And so
organelles.
right, these compartments are called organelles.
Yeah.
And so what we've done is, and we were deeply inspired by work from other people before us, is develop methods to break cells open but very gently, so all the compartments are still intact. We can isolate them, we can purify them if you want, and then we can take these purified compartments and use mass spec to ask which proteins are in which compartment.
Yep,
And
that's
if we do
exactly.
that
Yeah.
in a systematic manner across all the different compartments in a cell, we can get a complete spatial map of cellular organization.
That's amazing. That makes sense. Yeah, that kind of initial process is what I was thinking, where you get the organelles separated, get that mass spec data.
And
That's
if you want to get a visual understanding of what that looks like, Nicole actually made a fantastic little cartoon, which I really love, to really
Yeah.
show how the sausage is made there. Which is like, the idea is that we're pushing the cell or cells through a very small opening, an opening that's small enough so the cells have to break to go through, but the opening is big enough so that the internal compartments stay intact.
amazing.
And I just want to take this opportunity to thank you for being so open, Manu, to this style, because it's really fun to explore different ways of depicting things. And this is very different than I think a lot of standard science animation and illustration. It was much more playful. So I really enjoyed the opportunity to do that. Yeah. And I feel like in some instances like this, where you want to make it really clear, like this, we're representing the data, we're not showing you the data. You can go to the paper and see the incredible data there. Sometimes it's fun to use a more playful style and approach. So,
I was just gonna ask, yeah, with kind of this more detailed glimpse into your research, like the bigger organization of CZ Biohub and and your part in that. Like what's the dream? What's the big picture? Where do you hope to end up um down the line with all the work you're putting in?
Yeah, for sure. Well, there's a few different aspects to answering your question. Um, um, the Biohub is part of a bigger scientific project from the Chan Zuckerberg Initiative, who is doing a lot of work in the scientific ecosystem, with one clear mission, which is to cure, prevent, or manage all diseases by the end of the century. And it's a big ambitious goal. But if you look at where biology and medicine was 100 years ago, right, and you extrapolate that to 100 years from now or 75 years until the end of the century, you realize that so much is possible, especially given the accelerating pace of scientific discovery, technological innovation, so on and so forth, development of new therapies. So that is the big goal, okay? And within the Biohub ecosystem, which are a series of scientific institutes, really what we're trying to do in the next 10 years is really focusing on what we call understanding the mysteries of the cell. Because we believe that an essential part towards being able to understand and we hope cure and manage all the different diseases is to understand better how cells work, how they're built, what goes wrong with them molecularly when disease strikes. And as we get better and better at understanding all these more like fundamental parts, that's really going to unlock some new ideas about how to fix things that are going wrong during disease and how to develop new therapies and so on.
And that's one thing that I wanted to point out and ask you more about of your research is one thing that was incredible was seeing how the locations of proteins changed within the cell, within their compartments, organelles, when some exterior force happened. So in the example I'm thinking of, when a virus infected the cell, you see
100%.
proteins totally change position. And my understanding is that for the most part, and you kind of alluded to this earlier, the focus has been on quantity. So if something happens, if a cell gets sick, you look at what is upregulated, what there's more of, what there's less of, compared to this. Like it's kind of like a whole new paradigm to think about where things are going and what that means for function. What are they doing now?
Mhm.
Absolutely. Again, what we were really passionate about was to capture these global maps of entire cells to
Wow.
really understand how the cell is acting as an entire integrated system.
Uh-huh.
Um, and really, what we were after is trying to understand how that system gets rewired in different disease conditions. And you're absolutely right that the first disease that we looked at was infection with a virus, and a coronavirus in particular called OC43, one of the things that gives us like the seasonal cold. Which is, something we're all immune to, so it's fairly safe to use in the lab. Um,
Hmm.
and and you're absolutely right. It was the classical way that um, the most direct thing that we can measure in cells in terms of changes are how proteins or gene expression goes up or down. So changes at the level of abundance. And there's a lot of examples of that, and that tells us a lot of things. But now, for the first time, with the tools that we developed, we could also see how the internal organization of the cell was getting, again, rewired, completely modified, how different proteins were essentially getting relocalized to different parts of the cell during the disease. What's fascinating there is that they gave us a completely uh, complementary way of understanding all the different processes going on in the disease cell. And actually, in our case, gave us new ideas about how to fight that virus, which we absolutely would not have gotten if we were just looking at changes of abundance, proteins going up or down.
Really?
So what I think what we're looking at, and that might be kind of like a fundamental ways that cells use to change their function, is that there are two different modes of regulation that seem to be fairly orthogonal or independent from each other. Um, there's a regulation at the level of changing the abundance of things,
Okay.
and there is another mode which is changing the localization of things. And they're both equally important, and they're both giving us really fundamental insights about the mechanisms at play when cells get infected with a virus, for example. And both give us different ideas about how we could fight this process to cure the disease.
That's amazing. For in my head, the way I think about it is, yeah, for certain diseases with that localization piece, like, here's the hospital for this disease. Uh, yeah, that's incredible.
Yeah. I mean, and there's so many. So you can study the cell itself that's been infected. And then after that, I imagine you can study, say, the immune cells that are activated and see how they change. So
You're
what,
absolutely right. Would love to be able to do that.
yeah, what are the next things that you're excited to use this technique to look at?
Yeah. The next things are looking at different cell types because, so far we make it sound pretty simple. We talk about cells in general, but
Uh-huh.
really, I mean, it's amazing how many different kinds of cells exist in our bodies, and they're all different, and therefore they respond to disease or to perturbations very differently, and we know very little about that. Um, we know we know some general rules about how cells work and respond to perturbations, but specific rules about how specific cell types respond, for example, to the same perturbation, something we really, really poorly understand. And I think that's going to be one of the big focus of our science in the years to come and in the field in general, really. And so, for example, um, what's really cool is that we have more and more tools to recreate human biology in the lab. We can differentiate cells into specific cell types. I can take a piece of your skin and I can make a neuron out of it, or I can make a cardiac cell out of it. I can actually grow these cardiac cells, and literally they can start beating in a dish. And
That's
really recreating,
amazing.
yeah, it is amazing, recreating some fundamental physiology there. And so
Yeah.
we're super excited to use these new tools to really
Mhm.
be able to zoom in into some specific parts of human physiology, but still using the power of in vitro systems that we can use and modify and engineer to really be able to dissect molecular mechanisms with as much precision as we can.
That's really amazing to think about, and especially in light of the larger goal of CZ Biohub of curing disease. I mean, it is really incredible to think about, like, germ theory didn't even exist, not so long ago, and now we're to a point where we can differentiate cells and test, like, how they
I mean,
vary.
imagine, a century ago, antibiotics didn't exist, right?
Yeah.
Yeah.
And then we would die from a toothache, essentially. It's
Yeah.
just
Mhm.
incredible, right? And even in the last few years, now, immunotherapy is completely transforming the way we can treat cancer. So many cancers. I mean, when I was growing up, I remember some of my family members, right? Uh, 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 Being affected by cancers that are completely curable today.
Yeah.
And that was not so long ago, right? And so, again,
Mhm.
if you extrapolate that
Mhm.
development, I think we're going to get somewhere really exciting.
I did not expect to feel uplifted today, but this is really it's nice to have some good, hopeful things to talk about. I appreciate it. It's incredible.
There's
Um,
another aspect of why it's important to understand how cells are built is because more and more, I think, we're going to start using cells as devices that we want to control. There's a lot of work being done right now in engineering immune cells to really allow them to tackle some specific uh, diseases. Sorry, I'm not explaining that very well, but this
mhm,
idea of engineering cells uh, for therapeutic applications, but also for a lot of different biotechnological applications. Let me give you another example: detoxification. We all
mhm.
talk about uh, PFAS, for example, right? These forever chemicals. There's a lot of
Yes.
work that's being done right now in trying to engineer bacteria or yeast to break down these toxic chemicals. And frankly, some of these ideas look very promising. To be
Mhm.
able to engineer cells to perform new functions and really help us with some real world problems, we need to understand how cells are built. We need to understand how biochemical pathways can, break down molecules between different steps. And so that essential knowledge of cell biology is also going to be really the big enabler for cell engineering for biotechnology and helping us in the real world.
Yeah. I think that actually perfectly leads into something that has been going around. It's been kind of a trend right now, um, on the note of engineering. So, one a piece of your toolkit is the CRISPR, and I'm sure you've seen in the news recently that being used to, uh, what would be
Yeah,
the word? Engineer a
treat.
dire wolf? Bring it back?
Uh, yes.
So I'm curious what your thoughts are on that, like, used in a different way, not really for the sake of research itself.
Well, scientists are very creative, and they will come up with. Oh, and I'm not sure. I rewatched Jurassic Park not so long ago, so I'm not. su- Uh, I'm not sure exactly where we're going with some of these ideas.
Same.
But yeah. Um, but look, I mean, and I think this is part of a broader conversation. But of course, as our tools get more and more powerful, and what we can do with these tools becomes broader and broader, then um how careful we need to start to be with thinking about the applications and what that means for society as a whole is also an important discussion. um
Definitely. I think we're faced with that question in so many ways now, uh,
Mhm.
which is can be tough to navigate.
Yeah. Yeah. So I guess I'm curious, thinking about the incredible tools we do have now. There's so much opportunity, but it's also a really challenging time for a lot of scientists in the United States. And we don't need to get into those details today, but I'm curious if you have advice for young aspiring scientists who are, yeah, excited to continue pushing, what's
For sure.
possible.
Look, I can tell you that I feel like it's such an exciting time in biology and in cell biology in particular. I mean, we're
Hmm.
going, I think we're at the beginning of a revolution,
Mm.
and we have all the tools now in front of us to really understand how. I was talking to a colleague recently who was making the point that you think the universe is complicated, but when it's easy to do a back of the envelope calculation, which I will not try to do today, uh, and realize that actually what's happening inside cells is even more complicated than what's happening in the universe.
Uh-huh.
If you look at the number of interactions and all of that, it's
Mhm.
just mind-boggling. And this is why, I mean, life is such a fascinating
Yeah.
process, right? Um,
Uh-huh.
we have the tools today, both the experimental tools and the analytical tools, and of course, AI is a huge part of that, to really start disentangling the rules that make life possible.
Mhm.
And I think this is such an amazing opportunity for the new generation of scientists.
Hmm.
Yeah.
And it's so exciting to see so many students really drawn by biology these days in a way that that was not necessarily happening like 20 years ago. So um, I encourage every young scientist to take biology classes in college, realize the possibilities, the breadth of the questions that we can ask, the importance of it, because, as we were discussing, there are so many real world applications that are enabled by all of that, and so many different ways to make an impact.
Thank you. That's an incredible, yeah, inspirational message.
Yeah, it really is. So I have a couple just wrap up questions. I have two. So I'll tell you the two questions and then yeah, you can answer as you like. One, where did you grow up? And do you think that where you grew up and your upbringing kind of shaped the path you took and the way you look at science? And then two, if you could go back and give your 9-year-old self a piece of advice, what would that be?
Oh, that's such a good. That's such a great question. Well, I grew up in southern France. You probably heard some of that from my accent. And I mean, I think what shaped my path is maybe not so much where I grew up, but the people I met along the way, and especially my teachers. And I think, and of course, my parents, but the people that really mentored me into becoming the curious person that I ended up becoming. I don't think I would have done that but myself. um and so probably the best advice, basically the advice I give to a lot of my junior colleagues is, um take all the help that you can. embrace mentorship. Um, um, when I was younger, I thought that I had it. I had it all figured out, or I was gonna, come up with all the answers by myself because I'm this like super confident, like young person, and um, I embraced later the power of mentorship, the power of understanding that I'm actually ignorant about so many things, and it's so powerful to learn from other people. And so yeah, keep that in mind. Surround yourself by people that you enjoy working with and that you can learn from. And in my case, I mean, frankly, even that's still the case today. I learn so much from the people that I have the chance to work with. And just keeping that open mind. That would be my best advice.
I love that. I think, yeah, mentorship is just so crucial, and teachers, oh my gosh, we owe so much to teachers that we meet along the way.
Yeah,
And
yeah, it's so crucial. It's so much
Yeah.
fun also. It's just fun to work with people, to interact
yeah.
with them, to,
Hmm.
to share ideas.
Yeah.
Um,
Yeah. And I may have recently had a birthday, and
Mm.
it's making me realize
Happy
that is
belated
one nice
birthday.
thing. Thank you. That is one nice thing about getting older, is I feel like, getting to that point where you feel okay with admitting what you don't know
Yes,
and asking questions. It's
Yeah.
yeah, definitely something that I'm very grateful to be at that place in my life that I'm like, I'm okay admitting what I don't know and
I
trying to
love
learn
that.
more. And
Absolutely. Let's embrace
yeah.
all the things that we don't know.
Yes. Yeah, it's one of the great things about science too. So
Yeah, that's why we keep doing it.
mm,
And there's we're gonna be busy for a long time because
yeah.
there is a never-ending list of things that we don't know. And
Very true.
even things that we thought we knew,
Yeah.
now, evaluating them with like newer tools and
Yeah.
more precise technologies, we realize that, well, we actually had it all wrong. And so
Yeah,
that's
revisiting that stuff can be sometimes the most challenging part.
it.
Yeah.
Well, but that goes back to our
Rewriting.
conversation about having an open mind, right? And just, being reminded that, yeah, we are just scratching the surface all the time, and we're
Mhm.
wrong a lot of times.
Yeah.
It's okay to be wrong. Yeah,
It
let's
is.
realize let's realize when we don't know, and let's remember that it's okay to be wrong.
Yeah.
Yes. Yes. I think that's a great way to end our podcast.
All right. Thanks so much, both of you. It was fun to chat.
Manu, thank you so much for sharing your time and your story with us today.
Yeah,
It was
you're
such a
welcome.
pleasure.
Okay, bye
And
now.
just to all our listeners, thanks for tuning in. If you enjoyed this episode, give us a like, leave a comment with your thoughts or questions, and please hit that subscribe button so you don't miss more conversations